This video highlights some of the basic chemistry and cell biology underlying transfection. Nucleic acid delivery is an integral part of many researchers' daily experiments. Hopefully our discussion today of the basic mechanism of transfection will make your experience with transfection go more smoothly. Mirus takes great pride in being experts in the field of nucleic acid delivery. This stems from the fact that Mirus scientists have had a singular focus on transfection technology since 1995. Since then, the company has expanded its offerings to include chemical transfection, electroporation, in vivo delivery, and nucleic acid tracking products. So what does the term transfection mean? Interestingly, the term transfection was first used to describe the infection of a cell with viral nucleic acid. The term has evolved over time and today has come to mean mostly the delivery of exogenous nucleic acid into mammalian cells or insect cells. This is not limited to plasmid DNA, but it could be siRNA, microRNAs, CRISPR guide RNAs, oligonucleotides, or even proteins. Various methodologies are available to researchers who are performing transfections. The simplest and most popular approach is reagent-based transfection. The first chemical transfection reagent to be used was calcium phosphate, which could bind and form complexes with DNA. This method, despite being very cheap, suffers the drawback of being highly variable from one experiment to the next. Many commercially available reagents are based off of lipids, and this includes the use of cationic lipids as well as liposomes. Another major component found in transfection reagents is cationic polymers. Some examples are polyethyleneamine, or PEI, cationic proteins, highly branched compounds called dendrimers, and DEAE dextran. There are also instrument-based methods for nucleic acid delivery, the main one being electroporation, which uses an electrical pulse to create transient pores in the cell membrane through which nucleic acids can enter the cell. Other physical approaches include microinjection, bioballistic delivery, and the use of an externally applied magnetic field. Then, finally, some researchers also refer to virus-mediated techniques as transfection. Today's talk will focus on reagent-based transfection. We will first go through what we know about the basic mechanism of transfection, followed by an overview of the various readouts that can be used to evaluate transfection performance. Next, we will discuss the topic of cell health and how you can achieve the balance between the desired level of expression and any cytotoxicity that may be associated with the method. Finally, we will bring all of this together to give you some tools to optimize your transfections to suit your particular experiment. Chemical transfection has become popular largely because of the user-friendliness of the protocol. Here is a schematic to illustrate a typical DNA transfection workflow. First, plasmid DNA is diluted into serum-free medium such as OptiMim. Next, a transfection reagent is added to the mixture, mixed and allowed to incubate for 15 to 30 minutes. During this time, the transfection complexes are formed. Following this 15 to 30 minute incubation, the transfection complexes are added directly to cells growing in complete cell culture medium. Cells are incubated for a period of time that depends on the type of experiment that you are performing. Then, lastly, the cells can be harvested and analyzed. So not only is this a very easy protocol, it is amenable to high throughput, and no instrumentation is required except your routine cell culture supplies. It also affords decent transfection efficiency for routinely used cell types. So that's how you perform a transfection. Now, why do you need the reagent in the first place? The reason we need transfection reagents is because if you simply add nucleic acids to tissue culture cells, nothing happens. This is because mammalian cell membranes are inherently negatively charged, and this causes a repulsion between the negatively charged nucleic acid you intend to transfect and the negatively charged plasma membrane. So in this process, the role of the reagents, which are typically made up of cationic components, as we discussed, is to neutralize the negative charge on the nucleic acid and in some cases lead to condensation of the DNA in order for the complexes to be taken up by the cells. So then what exactly happens during transfection? Let's imagine a typical transfection where a cell is growing in tissue culture medium surrounded by transfection complexes. Some of the positively charged complexes will bind to the negatively charged membrane. Once bound, the complexes will be taken up by a variety of endocytic pathways. We know that clathrin and caveolin-dependent pathways play a role in the uptake of transfection complexes into the cell, but macropinocytosis and other mechanisms can play a role as well. So the first barrier to transfection is crossing the cell membrane. Once inside the cell, the next barrier to overcome is escape from the endosome. The nucleic acids must enter the cytoplasm. If you are delivering, for example, an siRNA or microRNA, this would be the end of the journey if the cytoplasm is the site of action. If you are delivering plasmid DNA, then you must also cross the nuclear envelope to gain access to the cellular transcription machinery. Nuclear entry, we believe, is largely a passive phenomenon that relies on nuclear envelope breakdown during mitosis. This is why it is important that your cells be actively dividing when you perform DNA transfections. Many transfection reagents are made up of cationic lipids that bear positively charged head groups on one end and long hydrophobic tails on the other. Examples include DOTAP and DDAB. The polar nature of these lipids allows the positively charged head group to associate with the negatively charged nucleic acid, while the hydrophobic tails can self-associate to form complexes called lipoplexes. Cationic lipids are often mixed with another class of lipids called helper lipids to increase transfection efficiency. Examples of common helper lipids include DOPE and cholesterol. Helper lipids, in conjunction with cationic lipids, can form structures called liposomes that can associate with and encapsulate DNA. These liposomes can be unilamellar or multilayered, and although not depicted here, it is also possible for the DNA to associate with the outside of the liposome. It is often seen that these liposomal formulations, while they can be an effective mode for gene delivery, are often more toxic than other formulations. In addition to lipid-based formulations, some transfection reagents utilize cationic polymers. These molecules neutralize the negative charge on nucleic acids and can be very effective for nucleic acid condensation. An example is polyethyleneamine, which can be linear or branched. Instead of the polar head groups found in cationic lipids, these compounds have positively charged groups, such as amines, which are present along the backbone and the side chains of the polymer. When combined with DNA, these transfection complexes are referred to as polyplexes. It is also possible to use lipids and cationic polymers that, when combined, work synergistically to bind and condense nucleic acids. Reagents that employ both lipids and polymers in conjunction with DNA are referred to as lipopolyplexes. Lipopolyplexic combinations are often the least toxic when compared to all of the other formulations, such as liposomes. Now that we have discussed the chemical makeup of transfection reagents, other features of transfection complexes to consider include the size of the complex, which in most cases ranges anywhere from 40 nanometers up to 1 micron, although most functional complexes typically fall within a tighter range, somewhere between 300 and 500 nanometers. Very small or very large complexes are generally not very efficient for transfection. Another key parameter impacting transfection efficiency is the overall charge of the transfection complexes. As I've already mentioned, regardless of formulation, the transfection complexes typically need to bear a net positive charge. But it is also important that this positive charge is exposed at the surface of the complex, where it is available to interact with the cellular membrane. This charge density is referred to as zeta potential. Another parameter related to charge that is important to emphasize is the charge ratio of the complex. This is referred to as the N/P ratio and refers to the ratio of the positively charged amine groups on a polymer to the negatively charged phosphate groups on the nucleic acid backbone. Manufacturers of transfection reagents typically recommend testing a range of ratios of microliters of reagent to micrograms of DNA, essentially optimizing the N/B charge ratio for your experiment. This graph shows the effect of titrating this ratio on the output of the transfection measured in luciferase activity. Here, 3 different reagents were tested at different ratios of reagent to DNA. As you can see, each of them has a different profile, but peaks somewhere between 3 to 4. This is something that varies from one cell type to the next, and in some cases has a very substantial impact on the overall transfection efficiency. So moving along now, let's just touch on a few ways that you can evaluate transfection performance. Most researchers performing transfections will typically rely on reporter systems for evaluating transfection performance. Certainly the most common reporter is green fluorescent protein, which allows you to directly visualize transfection and qualitatively assess transfection output on a fluorescence microscope. If you're looking for a more quantitative assessment, you can also measure GFP expression on a flow cytometer. Another popular readout is the luciferase assay. This technique is very popular because luciferase assays are highly sensitive and ensure a very wide dynamic range. In fact, this is an assay we rely heavily on at MIRIS because of the sensitivity of the assay allows us to compare different reagent formulations with more accuracy. A few others worth mentioning are beta-galactosidase and secreted alkaline phosphatase, or SEEP. Beta-gal gives a convenient way to qualitatively assess performance using the chromogenic substrate X-gal without the need for a fluorescence microscope. SEEP is useful if you desire a secreted reporter. If you're doing large batch cultures, such as would be used in protein manufacturing, this method only requires sampling the supernatant. You do not have to lyse all of the cells. So far, we've talked about indirect ways to measure transfection efficiency by using a reporter system, but it is also possible to track nucleic acid delivery directly. Doing this is very straightforward using reagents such as our Label It kits, which can covalently attach either fluorescent or other tags directly to the nucleic acid. These chemical labeling reagents are composed of 3 regions. The label is shown in green, which can be fluorescent or non-fluorescent. The linker, shown in yellow, which facilitates electrostatic interactions with the nucleic acids. And then there's the reactive alkylating group, shown in blue, that covalently attaches the label to any reactive heteroatom within the nucleic acids. The labeling protocol is also very straightforward. You just add the plasmid, for instance, to the Label It reagent, incubate for 1 hour at 37 degrees, and then purify away any unreacted dye. You then have covalently labeled plasmids that you can then transfect and visualize. An important consideration for labeling nucleic acids to be used in transfection experiments is ensuring that they retain their function. Very high label densities can first of all lead to overall fluorescence quenching if the fluorophores are too close together, and furthermore, they may affect the functionality of the nucleic acid you are delivering. In the case of plasmid DNA, this could possibly inhibit binding of the transcription machinery to the complex, or it could inhibit incorporation of an siRNA into an active RISC complex, for example. So to address this matter, we have carried out optimizations to determine the number of labels that can be present on a nucleic acid in order to afford decent signal for visualization without compromising function. This example shows Cas7 cells transfected with a rhodamine-labeled plasmid that encodes YFP. As you can see in these images, you can begin to detect red puncta at 1 hour post-transfection that are the fluorescently labeled complexes. At 4 hours post-transfection, this labeled DNA starts expressing nuclear YFP, and the expression increases at later time points of 8 hours and 24 hours. This implies that there are no substantial negative impacts on transcription or translation. In the case of siRNA, we have demonstrated that at the correct labeling density, you can achieve efficient knockdown as well. These are HeLa cells transfected with fluorescently labeled siRNA, which you can see as green dots in the cytoplasm. When we compare knockdown to an unlabeled siRNA, which achieves roughly 80% reduction in gene expression, there is essentially no change when the siRNA that was delivered is labeled with Cy3, Cy5 fluorescein, or rhodamine fluorophores. Earlier we alluded to the fact that transfection is a highly membrane-active phenomenon. This can result in negative effects on cell health, and in fact, many researchers have come to accept cytotoxicity as a given in their transfection experiments. Part of our focus at MIRIS has been developing transfection reagents that are highly efficient but do not exhibit the toxic effects seen with other reagents. So now let's go over a study we performed to try to understand more about how to balance toxicity with expression. Here is a series of images that illustrate the time-dependent and formulation-dependent effects on cell health using different reagents. These are HeLa cells that have been transfected with DNA using several different reagents. On the left is our Transit LT1 reagent, followed by Transit 2020. And in comparison is a liposomal formulation that is popularly used, called lipofectamine 2000. At the 8-hour time point, you really do not notice any differences in cell morphology with any of the reagents. The cells still look like they are healthy and adherent. However, as you progress to the 24-hour time point post-transfection, the lipofectamine formulation wreaks havoc on the cells. The cells are shriveled up and rounded and have detached from the plate compared to the healthy and nicely confluent-looking cells transfected with the transit formulations. This also correlates with the GFP efficiency, because although the cells transfected with lipofectamine may be expressing GFP, they are essentially lost from the experiment. So this just drives home the point that you want to achieve the experimental conditions where you can get maximal expression with the least perturbation to cell health. Apart from just determining the overt effects on cell morphology and GFP expression, there are additional available assays to quantitate cellular toxicity. One that we regularly utilize in our lab is the LDH, or lactate dehydrogenase assay. Lactate dehydrogenase, or LDH, is a cytosolic enzyme present in many different types of cells. When the plasma membrane is damaged, LDH is released into cell culture media. So indirectly, this measures the level of membrane damage caused by the transfection. Here you can see another dataset of HeLa cells that have been transfected with a luciferase plasmid. You can see expression and the corresponding tox values from the matching cell supernatants. This data corroborates what we just saw visually with the same three reagents. The lipopolyplexic formulations are giving high expression with low toxicity, toxicity being shown by the line graph, and expression being represented by the bar graphs. Whereas the liposomal formulation leads to higher toxicity, particularly at the 5:1 ratio, so close to 60% compared to less than 20% with the Transit LT1 and Transit 2020 formulations. We wanted to understand more mechanistic details about toxicity beyond the general effects on morphology, viability, and transfection efficiency. In order to investigate this process, we utilized a stress response array to look at changes in gene expression following transfection. Here, a non-coding plasmid was delivered to HeLa cells with the same reagents, either Transit LT1, Transit 2020, or lipofectamine 2000. And then we performed a qPCR-based assay to measure mRNA levels of 84 key genes involved in cytotoxicity at 8 and 24 hours post-transfection. The results were normalized to cells treated with only plasmid DNA. Alteration in gene expression was assessed using a twofold increase or decrease cutoff and a probability value of less than 0.05. Expression of 9 transcripts was significantly altered by lipofectamine treatment at the 8-hour time point, and 25 transcripts at the 24-hour time point, whereas fewer genes were affected by treating cells with the Transit LT1 or Transit 2020 complexes. For example, only 4 genes at the 8-hour time point and 9 transcripts at the 24-hour time point for Transit LT1. So a couple of things came out of this. First, it appears that cells exhibiting overt signs of toxicity also have greater changes in gene expression, both in terms of the number of genes affected and the magnitude of the fold change. Also, even though all of the cells looked healthy at that 8-hour time point, there were still significant changes in gene expression happening. This would be highly important to you if you happen to be working in HeLa cells and the gene you were interested in studying was represented in this list. That is why it is important to note that this is only one cell type and only 84 genes. This is not the whole genome. These results could change dramatically for a different cell type or with a different reagent, and there are definitely other cell biological changes going on that we did not measure here. Also notably, most of the transcripts that were differentially regulated by transfecting the cells with either plasmid DNA with Transit LT1 or plasmid DNA with Transit 2020 for both 8 and 24 hours were also differentially regulated with the plasmid DNA lipofectamine 2000 treatment. We also performed pathway analysis on these transcripts, and the highest numbers of transcripts fell into the categories of oxidative stress response and xenobiotic metabolism. So all of those experiments were with HeLa cells, but of course the story changes with each cell type. So here are 4 cell types, HepG2, THP1, NT2, and HUVEC with different toxicity and expression profiles. Trans-LT1 gives the most expression with low toxicity in HepG2, whereas THP1 cells, Transit 2020 is the only option. You get no expression with Transit-LT1, and lipofectamine only provides you with toxicity. For NT2 cells, the best formulation would be Transit-LT1, and in HUVEC, you would choose Transit 2020. These results demonstrate just how critical it is to identify which reagent formulation exhibits the most favorable expression and toxicity profiles for your cell type. Now moving on to ways that you can set up your experiments to get the best results. Going back to the workflow that we showed at the beginning of the talk, you can ensure success by taking a look at each of the components involved: the media you use for complex formation, the type and quality of the nucleic acid that you're transfecting, the amount of time that you allow complexes to form, the cell density at the time of transfection, and then also timing your downstream experiments such that you reach maximum expression or knockdown depending on what you are trying to achieve. One of the major factors impacting transfection efficiency is the composition of the serum-free medium used for complex formation. Serum is avoided at this step because it contains nucleases that can degrade the nucleic acid before it has had time to associate with the transfection reagent. Some antibiotics, such as kanamycin, are cationic and will inhibit transfection complex formation and should be excluded from the complex formation step. Similarly, some serum-free media contain polyanions, such as heparin or polydextran sulfate, that may inhibit formation of active transfection complexes. Certain transfection applications require surfactants, such as Pluronic for long-term growth and increased protein yields. However, these can inhibit transfections at high concentrations and should be avoided altogether during complex formation. The next critical component of the transfection workflow is the nucleic acid. Nucleic acids used for transfection should be highly pure and free of endotoxin. Ideally, DNA used for transfection should have a 260 to 280 ratio of 1.8. Endotoxin is a lipopolysaccharide present in the outer membrane of gram-negative bacteria such as E. coli. Many MaxiPrep kits will afford the desired level of purity. It is important to verify that your chosen plasmid prep kit states that it yields transfection-grade or transfection-ready DNA. The importance of avoiding endotoxin is illustrated in this bar graph. This data shows Cas7 cells transfected with a luciferase plasmid. When you are using very pure commercial-grade plasmid, you achieve high levels of expression compared to a crude plasmid prep containing high levels of endotoxin. If you take this same crude plasmid and remove the endotoxin using our MiraClean endotoxin removal kit, you can restore high expression levels. The features of the nucleic acid also play a critical role in transfection. Most of you are aware of using the appropriate promoters for maximal expression from plasmid DNA. But here we will consider features of messenger RNA. Although mRNA is less stable than DNA, it offers several advantages over DNA for certain transfection applications. These advantages include the fact that mRNA does not require nuclear localization for expression and that peak protein expression can be seen more rapidly than with DNA transfections. The presence of a cap structure or analog is important for expression, as is a poly-A tail. Most eukaryotic mRNAs have a 5' 7-methylguanosine cap that increases mRNA stability and functions in translation initiation. For this reason, mRNAs that will be used for transfection should either bear a cap analog, or an IRIS sequence to promote translation. Similarly, polyadenylation increases expression and stability and can be added to the mRNA either by using a poly-A polymerase or by encoding the poly-A tail in the DNA template. The nucleic acid dosage also matters in your transfection experiments. Here are 3 different cell types, Cas7, CHOK1, and HEC293, transfected with different dosages of DNA. What you can see is that a 2.5-fold increase in DNA dosage leads to higher expression, although there isn't necessarily a linear relationship, and the expression and the impact of dosage varies between cell types. Similarly, with siRNA, oftentimes very efficient knockdown can be achieved with low concentrations of siRNA. In the experiment shown here, you quickly reach maximal knockdown when adding increasing amounts of siRNA, right around 10 nanomolar. Beyond this, you're not getting any additional benefit and may trigger an immune response or off-target effects when using higher concentrations. For this reason, we generally suggest starting at 25 nanomolar for your siRNA experiments and adjusting from there. The amount of time that you allow transfection complexes to form is also critical in the transfection workflow. Most of our transfection protocols call for an incubation time of 15 to 30 minutes. During this time, the reagent is coating and condensing the nucleic acid, and the particles are growing in size. Here is an example of complex formation time compared to luciferase activity in CHO-K1 cells. Before the 15-minute time point, the complexes are too small for this time point, and beyond 30 minutes, the complexes are likely too large, and you start to see the efficiency begin to drop off. This effect is even more pronounced with mRNA, which requires a very short incubation time, only 5 or at maximum 10 minutes. This is one of the reasons you want to prepare your transfection complexes fresh with every experiment. It is not a good idea, for example, to form them the day before and store them at 4 degrees. Cell culture conditions such as confluency also play a role in transfection efficiency. Here's an image of 3T3 cells showing confluencies anywhere from 10% confluency to 90% confluency. Performing transfections on the very low end of confluency can lead to problems with cell health, whereas performing transfections on completely adherent cells can result in very low transfection efficiencies. To illustrate this point, here's a graph showing 3T3 cells transfected at 75% confluency and 100% confluency. The cells at 75% confluency show very high efficiencies, whereas the fully confluent cells, which are likely not dividing, do not transfect well. In situations where you are using suspension cells, the overall cell density matters as well, much like adherent cultures, although the effects may not be tied to contact inhibition. Using our Transit Pro transfection kit, which is optimized for protein production in suspension CHO and HEK cells, we have found that the optimal cell density is typically between 500,000 and 1 million cells per mil. So cell culture conditions, the rate of cell division, and the nutrients available in the cell growth medium are all factors that impact protein output. The last parameter that we will talk about optimizing is the harvest time, or when you actually do your downstream experiment following transfection. Of course, this is primarily determined by the goal of your experiment. For example, typical harvesting times for overexpression studies is between 24 and 72 hours. For gene knockdown, it is generally recommended to wait slightly longer, between 48 to 72 hours. This depends on the stability of your target mRNA, and if you're assessing knockdown at the protein level, the half-life of the protein plays a factor as well. mRNA, on the other hand, is very quick. You start to see expression at the 4-hour time point following transfection, and expression typically peaks within 24 hours. For specialized applications, such as therapeutic protein production, a longer time frame may be desirable, anywhere from 2 days to 2 weeks, to get the appropriate cellular biomass and the associated protein yield. For an example of this effect, here are some insect cell lines harvested at different time points, and you can see in each case there is an increase in the overall expression at longer incubation times. That concludes our discussion of how to optimize transfections, but if you would like to learn more about tailoring your experiment to particular transfection applications, I encourage you to explore our website, which offers a wealth of information on application areas such as virus production, genome editing, stem cell research, and more. One particular application that is becoming more common is co-transfection, and we recently released a product that delivers both DNA and small RNAs such as siRNA and microRNA, either independently or together in the same transfection complex. We also have customer feedback that this product will deliver guide RNAs for CRISPR experiments. Additionally, we have information on our site for setting up co-transfection experiments with various different types of nucleic acids. We also have a great resource on our site called Transfectopedia. This is essentially an online transfection textbook, so be sure to check this out as well. That concludes our discussion of transfection technology. If you would like to know more details about nucleic acid delivery, please visit our outreach page at www.mirasbio.com/outreach.